Welcome to the Woman's Connection. I'm Barrie-Louise Switzen, your moderator. The Woman's Connection is a program about events shaping women's lives and helping one gain authentic power on a personal or professional level. So won't you stay tuned? Welcome. Today we're going to talk about evolution. It is a topic that comes up periodically, and there's quite a bit of controversy about it. My guest is Dr. Lynn Caporale, and I would like to welcome her. Thank you for joining me.
Thank you for having me.
Darwin in the Genome. I have to tell you, reading it, I was intrigued, fascinated, and somewhat intimidated. How did you ever come to write the book?
Well, I felt, as a scientist, I felt that a lot of what we're learning now is so interesting, important, and relevant, actually, to everybody's life. And it's a shame that it's kind of hidden behind the language of technical jargon. And all we're learning about where we came from and how we're connected to each other and all of the medical breakthroughs that will come from understanding the genome.
Explain to me the difference, or maybe that they're similar, DNA and genome.
DNA is really the name, it's a nickname for a chemical.
All right.
And that chemical describes the chemical content of the genetic material. Actually, this chemical, it transmits information between generations. That's why you look like your parents, and it's because of the information carried between generations in the chemical DNA. Genome is the totality of the information that's carried by the DNA.
What is so important that people have spent 50 years studying this? And there's something also about the double helix?
Yes, well, actually they've been studying DNA for, it was originally discovered in the 1860s.
That's more than 50 years.
Yeah, but you're right, it's the 50th anniversary of discovery of the structure of DNA, the double helix that you mentioned. And so before then, DNA was found actually in the pus of wounds. And so it was studied by chemists as an interesting substance. We're interested in substances associated with life. They determined its structure, they named it. But it was thought to be a rather boring molecule. It was just made up of 4 things. And even though eventually we learned it was in the chromosomes that we know are important for heredity, because it had this kind of just made up of 4 things, scientists thought it couldn't possibly be responsible for transmitting the information from generation to generation.
How did they finally realize that this was important?
Well, actually it was here in New York at Rockefeller University. Somebody was studying, a man named Oswald Avery was studying a very interesting phenomenon with pneumonia. The bacteria responsible for pneumonia, there was a kind of benign form of it that could infect you but not give you pneumonia, and it wasn't virulent. And then there was this very dangerous form. And he found that, or others had found, that if you actually kill the dangerous form, heat it, grind it up, mix it with soap, and then just pour what you get out of it onto the benign form, the harmless form of the bacteria, it would suddenly turn deadly and transmit that information from generation to generation. So you'd suddenly have a deadly. So they were curious, what's in this soup of the extracted killed bacteria that made it so deadly? And it turned out to be this previously thought to be boring molecule, DNA, that was transferring the information. And in fact, Avery himself wrote to his brother, who would have thought it? So it was a big surprise. Once, that happened in the 40s, and once DNA was recognized to be the genetic material, then everybody was excited to figure out how it transmitted the information. And that's why there was the race, some called it a race, others just scientific curiosity to figure out the structure of DNA.
Well, it's always you want to be the first to get the prize and be known for it.
Well, that's what Watson and Crick did, and others like Rosalind Franklin was interested in the science itself. She was curious about figuring it out. She didn't realize there was a race on. She just wanted to know.
Who is Rosalind Franklin?
Rosalind Franklin was a extremely bright woman. She's born in England, studied physics, and made some and worked in a field of science called crystallography, where actually the shadows that we shine X-rays on molecules and look at their shadows. And just as by looking at our shadow, you might be able to reconstruct what we look like from the kind of pattern. It's kind of a diffraction pattern of the molecules. These scientists could calculate back, it wasn't straightforward, what the molecule was that created the shadow. So Franklin was an expert. She was brilliant. She made some of the most beautiful pictures of X-ray pictures of molecules and determined their structure. With DNA was to take the crucial picture that really revealed to those who knew how to interpret the pictures what the structure of DNA was. Now, unfortunately, she, who was a very calm, deliberate, careful worker, didn't really have time to report her results because
what a shame.
Somebody showed her picture to Watson, who immediately ran off and told Crick about it, and they rushed to publish their structure. Yes, that's what happened. Women in science, Rosalind Franklin is a particular heroine of theirs because they face a lot of the same being pushed aside often.
How did you get into this field in the first place?
Well, it's very interesting. I really hated science up through, I'd say, the 7th grade. It was memorized. To me, it seemed, I was wrong, that it's just memorizing boring lists of things. But in the 9th grade, at that time, they don't do it now, they passed a carburetor around the room, and I stopped to try to figure out where the air went in, and just stopped to look at it for a second. And amazingly, a teacher of mine, I still remember her name was Miss Hemmer, spotted me in a class of 36 kids looking at this and decided that I was a scientist because I was curious. And before I knew it, me and I was with another little girl, Joni Goldstein, and we were in the science fair, and that sort of ridded me of my antipathy to science. I began to see that it was interesting, you could figure things out, you could think about it, but the road was not over. I enjoyed learning about science after that, but I still had no intention of becoming a scientist. And it was additional experiences with really good teachers, plus reading an important book, Death Be Not Proud, by John Gunther, about his son who died of a brain tumor, that got me interested in medical research.
Now, what kind of obstacles have you had to overcome in the scientific field?
There were a lot of them. I think, first of all, in high school, I know one of my classmates asked me why I was studying science, since I was just going to be at home and be a mother, and why work so hard in school. And I said, well, if my children ask me why the sky's blue, I can tell them. And when I went into college, and I was very interested in, I had begun to think about going into medical research. Again, my counselor, the guidance counselor who has to approve your program, dissuaded me from taking calculus, which was required for a chemistry major, because she said it would be hard. And why it would be harder for me than anybody else, I didn't know.
Because you were a woman.
Well, that was, that was the assumption. But fortunately, I had a little calculus one summer, and I was not intimidated by it, and I had the confidence.
Where did you get the confidence to push ahead in this field?
Well, I think I knew from classes that I was talking after class with the guys and explaining things to them. So I didn't feel that at least in an informal sense, that I was less intelligent or less capable. It was hard. It was hard work. You do have to study hard. And I think that's where a lot of women fall down, because they're told they can't do it, they find it difficult, and they begin to believe it, when in fact it's just a matter of
persevering. Would you explain what is important about your book?
Yes, I think I tried to do two things in the book. I tried to do something similar to what Darwin did, to write a serious scientific book that would stimulate scientific discussion, well referenced with a lot of evidence. And
that
yet,
you did. What is the argument in your book?
Okay, the main thing is, it's very hard, even for those who are completely convinced that evolution took place the way Darwin said, through natural selection, that it's just hard to conceive of how we get such complex life forms through a random mutation. and followed by selection. But in fact, what I say is that Darwin wrote, if you think about it, before we knew about DNA, before we knew it was the genetic material, he never said mutations were completely random. What he emphasized was the generation of variation, that there was a lot of variation within a species. You can see that dogs are very different from each other, but naturalists can see even within a species of beetle that different individuals within the species are different because they collect them, and so they recognize that. So the idea that there's tremendous variation and selection acts on the variation. So what is different about my book is that I point out that the mechanisms that generate this variation can fall under selective pressure, just as wings and beaks do. So what do I mean? I
was going to say, what do you mean?
What do I mean? Yes. What I mean is basically that living things can get better at evolving, just as wings can get better at flying.
So do you think we've gotten better?
Yeah, well, we can get better at evolving. So, for example...
Well, wait a minute. How do we get better from evolving?
No, what I'm saying is that life gets better at generating the variation at which selection acts. So let me give you an example.
Okay.
And not for people, because it's much harder to visualize than people. Let's look at bacteria. It's another life form. It's been here a long time, and it's reasonable to think that we've learned what the bacteria have learned. They have certain genes that tend to mutate more quickly than other genes. So in other words, selection has focused them into generating variation just where they hit us the worst. The genes that are involved in there sticking to us, and also genes that enable them to take up information from the outside and incorporate it into their own DNA so they can learn new things more rapidly. So all of this is better than random.
So they're almost like a sponge.
Well, they're in a way they learn.
Okay.
They learn. So they are not limited to just randomly trying things until they finally get something that works. And that's similar in our immune system, and I'm arguing it's similar in the whole genome. As the genome explores new forms, it focuses on those areas of exploration where it has that are most likely to be generate protection or new skills.
Self-preservation.
Yeah, they're more. For example, our antibodies. We know we all make antibodies against bacteria or viruses that infect us,
Like scars or HIV, AIDS, and stuff like that.
yeah, and pneumonia and measles and all the things and so when we make antibodies, our body is learning new structures. It's actually changing its DNA in the cells that are going to make the antibodies. But it doesn't randomly change all the DNA, because that's not what it needs. It just needs to change a little piece of the DNA that is responsible for encoding the part of the antibody that's going to stick to the bacteria. So it focuses change right there where it needs it. And it's not all random mutation. That's
not.
No, that mutation, that through generations, eons, life learns about the world through selection.
So what you're saying, through DNA, that you and I are connected. If we looked at your DNA and my DNA, we'd have similarities.
It'd be actually 99.97% approximately the same. That's like rounding error, right? It sounds like rounding error. So all humans are that similar to each other.
If that's the case, then how come there's so much controversy about evolution?
Well, I think, when people think about evolution, they immediately go to the idea of monkeys and, seeing us evolving from monkeys. And
You think that we have evolved from monkeys?
yes, but not monkeys as we see them today. We've evolved from the same thing the monkeys we see today
Evolved from.
So evolution, so some people can't accept that But the evidence is so strong, and it's important to study evolution because we think about monkeys, and when we discuss it, usually in controversy, Scopes monkey trial. But in fact, evolution takes place in bacteria in our bodies very, very quickly. And there's a recent case in the Netherlands where an infant actually evolved an antibiotic-resistant bacteria because one bacteria in its body that was harmless but had some antibiotic resistance information passed it over to a very dangerous bacteria that it had. And so this little 2-month-old infant had right within it the evolution of a dangerous strain of bacteria and was fighting for its life. And so it's important to understand evolution. To understand medicine, it's important to understand evolution. Certainly to understand infectious diseases. We've recently seen the SARS epidemic. Where did it come from? Those are questions about evolution, actually. Where did this new pathogen come from?
What I found fascinating was the fact that when you were talking about the different DNA elements, I know you're going to correct me if I'm wrong, so please jump right in. But you were saying that they were all different colors.
Used colors as a way of looking at them, rather than chemical structures, which I thought would be intimidating and not necessary really for the discussion in the book.
Oh, so wait a minute. So in other words, what you're saying is that DNA is all black and white?
Well, it absorbs in the ultraviolet range. So if you had DNA, actually it's a beautiful molecule. It's a kind of a translucent, fibrous molecule you can roll up onto like a glass rod, and it looks translucent, and it catches like shimmering. It's beautiful.
Everything has got DNA.
Essentially every cell in your body, except your red blood cells, have DNA in them.
The red cells don't have DNA.
Well, they're kind of an end-stage cell, so the precursors, the cells they come from, have DNA. But then they've lost it because they don't need to divide. They're not going to stay for very long.
How does this play off in the drug industry?
Well, knowing the genome sequence is an amazing, will have a dramatic effect on drug discovery. There's a few ways that it's important. One way is just the ability to look at the big picture. Up until now, a scientist, say a biochemist like me, would think, well, if I block this particular protein, then we can treat this particular disease. If we block an enzyme that makes cholesterol, we could lower cholesterol. It's an example of an approach to drug discovery. But the problem is it depended on the biochemist kind of understanding things and knowing the exactly right enzyme to pick. But now we can look at the whole picture because we know we have a way of testing for all the genes and what's turned on where, and so we can begin to discover that proteins we didn't even know exist have an important role in blocking death of a cancer cell, for example, and begin to work around it. So it's really exciting for deepening our understanding of biology. In addition, it will enable us to target medicines more specifically to people who they will help and not people who either won't be helped or would be hurt. Because, often a medicine will help, say, 80% of the people and hurt 5% of the people you give it to, but we don't know who they are. Now we will be able to identify small genetic differences that are responsible for some of those.
It almost sounds like before a doctor prescribes a medication for you, you've got to give a DNA sample, so that can go back to the manufacturer to see if there's a problem with it.
Well, eventually there will be some, just as you have tests in the doctor's office now, there will be, with time, not tomorrow, but tests associated with some medicines. And there are already, but they're very complicated. But there'll be quicker and easier tests to say that this medicine will help this patient and not that patient. One of the places we're seeing that begin very seriously is in the cancer area, because cancer, we've been giving people toxic drugs, as you know, they're very toxic.
Yes.
And yet they don't always help. So here you have a situation where you're actually poisoning people in the because you know you're helping a certain percentage of them. So you want to do it, and people accept it. But
They
if
don't
we
have any
could,
alternatives.
but if we could figure out who that drug would help, or that combination of drugs will help, and who it won't help, that would make a tremendous difference.
In your book, you talk about the double helix. Would you explain it a little bit more?
Yes, it's a beautiful structure. Actually, that's what scientists all immediately say, it's beautiful. And why is it beautiful? Because looking at it, you understand immediately how it encodes information. It's made up of 2 strands, and the strands, just by looking at one strand, you can predict the sequence of the other strands. A string of letters, we call them A, T, G, C, and the order of the letters actually encodes the information. So, for example, if you have T, 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 that means you're going to put a particular amino acid into a protein. That's the order of the letter. If you have G, 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 another amino acid goes into the protein. So just the string of letters T, 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 G, 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 and so on, it will define the order of amino acids that make up our proteins, as one example of the kind of information. So, but the beautiful thing about the structure is you have this string of letters on one strand. You can immediately predict the sequence of letters on the other strand. Now, what does that mean?
A good question, because I have no idea.
What that means is when it comes time to copy the DNA
Right.
and send it off to another generation, the 2 strands can separate, and each one can be copied using that information. The information is in each strand is enough to specify the second strand.
So in other words, what we're doing is taking all of my DNA, putting it into my child and mixing it with my husband's DNA. Is that what you're saying?
Well, what you're doing actually is you take part of your DNA, because you have DNA from both of your parents,
All right.
and your husband has DNA from both of his parents. And your child can't have all that DNA, because that would mean that at each generation the amount of DNA doubles. So each of you has to pick. I mean, you don't personally mentally pick, but actually and when your eggs are being formed,
Yeah.
they're picking each egg, whether chromosome 1 comes from grandma or grandpa, and chromosome 2 comes from grandma or grandpa, chromosome 3 comes from grandma and grandpa. And the same thing is going on when the husband is forming the sperm, so that you get your children get half of the information in your DNA, and half of your husband's, and together you make a new child. Yeah, and
Fascinating.
and that brings us to the connection between all people on Earth are so closely connected, because if you think about that, that's at each generation half. So after 10 generations, how much of your DNA is in any individual? It's really, it's mixed in with the DNA of so many people who are around today whom you don't know. You may see them on TV clinging to a tree in a flood. You might have bumped into them.
That's not funny.
Yeah, but it's a sense of connection. So you may have bumped into them coming out of the subway. I mean, these are all people who will be your relatives 10 generations from now. And the other thing is to look 10 generations back in time, 10 generations ago. Again, your DNA came in little pieces from all of these many people, all of whom also gave DNA to many other people. So that makes us all very highly connected. And that's where you see this 99.97%
That's
identity.
awesome. So so your 99.97 DNA is identical to mine.
Right. Yeah, so it's only this little 0.03% that's different. And when you think about it, I mean, we're even around 95%, 98%, the numbers vary, identical to chimpanzees, who are our closest relatives. And even there are so many of our genes that you find in yeast and frogs. And why is that? Well, when you think about, let's think about a frog, for example. Now, to us, frogs look very different.
I haven't seen a frog really since I was dissecting it in biology.
Right.
Okay.
But look, the point is that we are so highly related to all life on Earth. We all use the same genetic material or very similar genetic material to carry the information between generations. We all use similar energy sources. We all have to maintain our genetic information and transfer it to the next generation. So we are really highly related to
Wow.
all and connected to all life on Earth.
What do you see going forward?
Well, there are two directions. One is the medical direction. There's just going to be a tremendous transformation in our ability to understand what we're doing. It's as if blindfolds were taken off. We were kind of going around blindfolded, really, trying to figure out what's going on in so many situations. Now we have the tools to begin to really look inside and pick up the hood and see what's going on in cancer and heart failure, everything.
Sounds exciting time to be alive.
It's a wonderful time to be alive. It's an amazing moment in human intellectual history that we have looked inside us and find the information that encodes what we become. The second part is that as people begin to understand this information, I would like to think that as it breaks out from the scientific community, we're all excited about what this sequence means or that piece of DNA, the sense of connection of all life on Earth, the preciousness of all life on Earth, and the beauty of how life could have evolved and come to be what it is on Earth today will penetrate, hopefully, beyond the borders of science and begin to be appreciated by people who are otherwise missing out, really, on so much about who they are, where they came from, and what the future is.
Thank you so much for
Thank
joining
you.
me. It's been wonderful having you and learning so much about the genome, DNA, and evolution. Thank you for joining us. I hope Dr. Lynn Caporale has answered your questions about evolution. Bye now.